Photosynthesis is a metabolic pathway consisting of light-dependent, needing light, and light-independent, not needing light reactions. Overall, water and carbon dioxide are used to produce carbohydrates, with oxygen as a waste product. The energy found in photons of light from the sun is used to produce ATP. The energy is trapped by the pigment chlorophyll in the thylakoid membrane. It is then used to break the bonds in the water molecule, splitting it into O2 and H+ ions. This is called photolysis. Photophosphorylation also happens. The light-independent reactions use the chemical energy stored in ATP and NADPH plus H+. This takes place in the stroma. The carbon found in CO2 is fixed in the form of sugars. Light energy from the sun is converted into chemical energy. This is trapped in the chlorophyll, which are grouped into structures called photosystems. The photosystems are found in the thylakoid membrane. When light hits the chlorophyll, electrons get excited and are lost in oxidation. The first step is photoactivation of photosystem 1. When an electron is energized by absorption of light, it is ejected from the photosystem and travels to the reaction center. The electron then passes down through an electron transport system and eventually back to the photosystem. The non-cyclic photophosphorylation can be broken into steps. Photons of visible light are absorbed by the pigment chlorophyll. Light energy raises the energy levels of electrons. This is called photoactivation. The chlorophyll is oxidized when two electrons move to the electron transport system. When water is split through photolysis, electrons are given to the photosystem. This is the source of H+ ions and the waste, which is oxygen. Photosystem 2 is then reduced so that it can absorb more light. The electrons are accepted by the electron acceptor and travel to the protein embedded in the thylakoid membrane. This is called the electron transport system. The electrons can travel to the protein pumps, and the protein pumps are reduced to pump H+ ions into the thylakoid space. If not, the electrons travel to photosystem 1. This is called photoactivation of photosystem 1. At photosystem 1, a different light frequency is absorbed. The photosystem is oxidized to release electrons. This is the same as what happens at photosystem 2. The next step is called reduction. Electron passes from photosystem 1 to ferredoxin, which reduces NADPH plus to NADPH plus H+. The NADPH stays in the stroma to be used in the light-independent reaction. Photosystem 1 is then reduced by electrons from photosystem 2. This is a graph showing the different energy levels. As the electron passes through the system, it loses energy. Since there is a high concentration of H+ ions in the thylakoid space, they can diffuse back into the stroma through the pore in ATP synthase. This process drives the photophosphorylation of ADP to ATP. During these redox reactions in a light-dependent reaction, the energy levels of the electrons change, as shown before in the graph. A reaction called the Calvin cycle takes place in the stroma and is controlled by enzymes. In carbon fixation, the single carbon in carbon dioxide is trapped by the ribulose biphosphate, a 5-carbon molecule. This forms two molecules of glycerate 3-phosphate, or GP for short. In reduction, GP is reduced to a triose phosphate, or TP for short, in the process which NADPH and ATP are oxidized to provide the energy. TP is used to manufacture a variety of organic molecules including glucose phosphate and other examples like sugar, starch and amino acids. This is followed by regeneration. Some of the TP is used to regenerate ribulose biphosphate. This will allow more carbon dioxide to be fixed in the atmosphere. This is just a summary. Ribulose biphosphate can also be called ribisco. The membrane is permeable to CO2, O2, ATP, sugar, and other products of photosynthesis. The rate of photosynthesis is measured at different wavelengths. The maximum rate are blue and red of the visible spectrum. The lowest rates are yellow and green. Chlorophyll absorbing blue and red light well, but not green. Using a calorimeter, different wavelengths of light are passed through and absorption is measured. This absorption spectra of chlorophyll shows the absorption of blue and red light and the reflection or transmission of green light. The rate of photosynthesis increases as light intensity increases. Photosynthetic rate reaches a plateau at high levels of light because photosystems are absorbing photons at a maximum rate. Photosynthetic rate reaches a plateau at high levels of CO2 because ribulose biphosphate carboxylase is fixing carbon at a maximum rate. The rate of photosynthesis increases with increase in temperature because greater kinetic energy creates more collisions between enzymes and substrates. High temperatures reduce the rate of photosynthesis because it denatures enzymes by breaking intermolecular forces which alters their active sites. Thank you for watching.